Good afternoon, guys. This is the lecture on DNA replication, the copying of DNA, and we're going to cover the information from pages 261 to 264 in your textbook. So you already know the basic concept of DNA replication. DNA is a double-stranded molecule, and each strand is held together to the other strand by hydrogen bonds in between the base pairs. Adenine always pairing with thymine, guanine always pairing with cytosine. And so the basic idea is that if we're going to copy, we're going to do that in a semi-conservative way, which means the left strand and the right-hand strand are going to unzip from each other. There's an enzyme called helicase that does that. It separates the two original strands. And then there's an enzyme called DNA polymerase that's going to build a brand new strand here that is complementary to this old strand. And it's going to also build a new strand over here that's complementary to this strand. And so it's pretty simple. If there's an A on the old strand, we're going to put a T on the new strand. C pairs with G, T pairs with A pairs with T. And when you're finished copying both sides, this is what you end up with here. You end up with two DNA molecules. Each one is an exact replica of the original, assuming that everything goes right. But each DNA molecule contains one old strand and one new strand. One old strand and one new strand. And so what we're going to do is go through the process in a pretty detailed fashion and talk about all of the different enzymes that play a role in this copying process. Okay, so the whole time that we're talking about replication today, it's important that you remember what we discussed last time about how each strand of DNA has a 5' end and then at the other end a 3' end. And the two strands of DNA always run anti-parallel. So if this strand runs 5 to 3, the other side of the DNA would go the opposite way. It would go from 3' to 5'. And the reason that's important is because when a new strand of DNA is made, the new strand is always going to be built in a 5' to 3' direction. And whenever we want to add a nucleotide, if we wanted to make this strand of DNA longer and we wanted to add a nucleotide to it, you always must add to the 3' end. This number 3 carbon is the one that would bond to the next nucleotide and the next nucleotide and the next nucleotide. We're always going to add to the 3' end. And the reason is basically that the enzyme DNA polymerase only recognizes a 3' end. It is unable to add anything to the 5' end. So that may not completely make sense to you yet, but just keep it in mind because it's really important. A new nucleotide is always added to the 3' end and can only be added to the 3' end. Okay, so this is what we're going to be diving into. You can see that DNA replication is more complicated than it seems like it needs to be. We're looking at a double-stranded DNA molecule here that is in the process of being unzipped, you might say. The two strands of DNA are being separated. So there's a strand of DNA that we want to copy up here, and then the other half of the DNA, the other strand, is down here. But you'll notice I've highlighted some of the key components here. Everything that I highlighted in yellow is either an enzyme or at least a protein. Most of them are enzymes. And then we've got these single-stranded binding proteins that I don't know if you would technically consider them to be enzymes, but they're all proteins. These are all proteins that play an important role in the copying of DNA. And this is a really nice illustration from your textbook, but it's a little hard to really figure out, I think, what's happening in this picture. And so to actually study the process of replication, I actually drew out a simpler picture with A's and G's and C's and T's so that we can look at it in a slightly more simplistic fashion, but still get the main concepts. So I've drawn in a simple depiction of a DNA double helix, and of course it's not twisted in its spiral staircase shape. I've simplified it by straightening it out here. But we can see we've got A, G, C, T, and so forth on one side, and then we've got the complementary bases on the other side. Notice the hydrogen bonds in between the A's and the T's and the G's and the C's. These little dotted lines are the hydrogen bonds. And notice both sides of the DNA, each strand of DNA has a 3' end and a 5' end. 3', 5'. So those always point in opposite directions, as we've talked about before. Okay, so I've broken this process down into a 7-step process. And for each step, if there is an enzyme or a protein that's in charge of that step, I've highlighted that. So anything you see that's highlighted in yellow is an enzyme or at least a protein. So the first step involves an enzyme called helicase, and it's named helicase because its job is to unzip or dismantle the helix. It literally separates the two strands of DNA. So helicase untwists and unzips a small section of the double helix. That's going to create what's called a replication fork. I'll show you that in just a moment. And then step 2, single-stranded binding proteins come in, and their job is to hold the two single strands of DNA apart so that they don't just flop back together. If you don't hold them apart, they will simply zip back together and the copying process will not happen. And so we're gonna talk about the first 3 steps kind of all at once. The third step is an enzyme called primase. It's called primase because its job is to create a molecule called primer. Primase is the enzyme that makes primer. What is primer? Primer is a small single-stranded stretch of RNA. It's not DNA, it's actually single-stranded RNA, but it's a little piece of RNA that has both a 5' and a 3' end. And the purpose of primer is to create a 3' end that the main copying enzyme, DNA polymerase, can latch onto and start building DNA. So none of this makes any sense without looking at a picture. So let's go ahead. We're going to take this DNA molecule, and what you're going to see is we're going to unzip it, almost like unzipping a coat, but we're going to unzip it from the bottom up. We're going to unzip this bottom area here, and we're going to create a replication fork. So notice I left out all these letters, but they're still there. I just left them out because we're going to focus on this area down here. We've unzipped just the bottom of the DNA. And now we've got a single strand that is no longer connected to this single strand. Those two single strands are being held apart by single-stranded binding proteins that we don't see in the picture because I don't have the artistic talent to try to draw all of the enzymes. But if we're going to build a new strand of DNA here and a new strand of DNA here, we can't just build DNA. We have to have something to latch onto. The enzyme DNA polymerase needs a foundation from which to build. And that enzyme only recognizes a 3' end. So the enzyme primase comes in and creates a little piece of RNA right here. A little piece of RNA right here. Those pieces of RNA are called primer. And they're not really this short. This is way oversimplified. It would certainly be longer than just one nucleotide. Um, but just to keep things simple, I wanted to show you this is just a little short piece of RNA. And just like DNA, each little piece of RNA has a 5' end, which this would be a 5' end right here, and a 3' end, which I labeled. So here's the 3' end, here's the 5' end. Okay. Even this little piece of RNA has a 5' end and a 3' end, just like the DNA has a 3' and a 5'. 3', 5'. Okay, so it's the 3' that we're interested in, because in the next step, DNA polymerase is going to come in and latch on to this 3' end and build a new strip of DNA right down here. And on the other side, it's going to latch on to this 3' end and build DNA in this direction. So this is a close-up view of what's going on so far. Here's our helicase enzyme. It's almost literally like the little plastic thing that slides up and down a zipper. If it slides in this direction from right to left, it's going to unzip the DNA as it goes. Once the DNA has been unzipped, you have these two single strands of DNA that are held apart by single-stranded binding proteins. There's one here, there's one here. And then in order to start building new DNA here and here, we're going to have a little piece of RNA primer. Notice that little piece of RNA has a 5' end, which we cannot add to, but it also has a 3' end that we can add to. So now let's do step 4. DNA polymerase, which I may sometimes refer to as DNA poly for short. DNA polymerase, DNA poly, begins adding nucleotides, A's and G's and C's and T's, in a 5' to 3' direction. Remember, new nucleotides can only be added to a free 3' end. The two parent strands of DNA are used as templates or patterns to determine what the letters are going to be on the new complementary strands of DNA. So here's that same picture. This is where we had our 3' end on this little piece of primer. So DNA polymerase is going to start right here, and it's going to read a T and put down an A, read a C, put down a G, G, C, A, T, A, T. So it's going from the 5' to 3' direction, but it started right here at this 3' end, and it worked its way down. On the other side, remember, this is the 5' here, so this was a 3' end right where my pointer is. So we're going to start at a 3' and go up this way from 5' to 3'. So it's going to read a T, put down an A, read a C, put down a G, G, C, A, T, A, T. And that's as far as it can go. Right now, we've done the first 4 steps of replication, but we can't go any further because the rest of the DNA is still zipped. So basically what's going to happen is helicase is going to continue moving up, and it's going to unzip just this next little section. Okay, so let's look at that. We're going to repeat steps 1 through 4 as we replicate the next little section. But first we have to unzip that next little section. So here's where we left off before. Helicase has now moved up a little bit, and we've now unzipped the next little piece of DNA. And single-stranded binding proteins are holding these apart still. And primase, now let me point something out that's very important to notice here. Now that we've unzipped the next little section, on the right-hand side, we have a 3' end right where we left off. This is the 5' right here. So this is a 3'. And that's good. We can add to a 3', and we can just keep on building DNA. But right here, look carefully, this is a 5' end. See, 3' here, 5' here. We cannot add to that 5'. So on this side, we have to bring primase back in to put another piece of primer here to create another 3' end right here where my pointer is. We don't have to do that on this side. We've already got a 3' end. But on this side, we have to add another piece of primer to create a 3' end, and then DNA polymerase will come in right here and go boop, 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 and build a little strip right here of DNA, and then it'll come over here and add on to that 3' end, C, G, A. Boom. That's what it looks like. But you're always going to have that same issue on this side here, which I'm now calling the leading strand. You're always going to finish off with a 3' end. The last letter is always going to be a 3' end on this side. But on this side, which I'm now calling the lagging strand, the 3' is way down here. So there's always going to be a 5' where you leave off, where the process ends. And so it's almost as if this side kind of lags behind. There's an extra step on this side. We're always going to have to add a piece of primer and then build DNA back down. Add a piece of primer, build DNA back down. When we unzip the next little section, we're going to have to add a piece of primer and then work our way back down on this side. And we will not have to do that on the leading strand because, see, it already has a 3' end. Now, what that does, it means on the leading strand, you have a nice, smooth, continuous piece of DNA that's uninterrupted. But on the lagging strand, you have a fragment of DNA and then a little piece of RNA primer, a fragment of DNA, another piece of primer. You're always going to have sort of fragmented DNA segments on the lagging strand. And the molecular biologist who discovered these little fragments of DNA was named Okazaki. So these are called Okazaki fragments. Okay, so we're going to continue repeating steps 1 through 4 as the helicase moves up and unzips the next little section, and then we'll replicate here, and then we'll unzip the next little section. Helicase just keeps moving up as this process makes its way all the way up to the end of the DNA. And so let me make sure I didn't skip anything. So I'm kind of skipping to the end here. This is what we would end up with. So here was what we were calling the leading strand. It has its little original piece of primer that we put on there at the beginning, and then the rest of this is just smooth, continuous DNA, 3' end here. But on this side, remember, we had to add primer and build DNA down. Add primer, work our way down. Add primer, work our way down. Add primer, work our way down. So we have Okazaki fragments on the lagging strand. Well, we have to get rid of those. Okay, so step 6 of the replication process is we need to get rid of these little pieces of primer and replace, remember primer, these little black areas, that's RNA. So we got to get rid of that. We got to replace it with actual DNA and join these Okazaki fragments together so that this side will be just as smooth as this side. And so step 6 is where DNA polymerase, the enzyme DNA polymerase, comes back in and replaces this little piece of RNA with DNA. And I guess I could sort of try to show that. I'm not very good at drawing on this thing. Let's see if it'll let me. So if I come in here and replace that RNA piece with DNA, replace this RNA with DNA. I'm doing a messy job, but you get the idea. Whoa, replace the RNA with DNA. Now, when that happens, when those little pieces of RNA are replaced with DNA, there's still going to be a tiny little gap. Like in this case, there would be a tiny little gap right about here in between this guanine and this adenine. There would also be a tiny little gap right here between this adenine and this adenine. And so there's an enzyme that works almost like a welder. It has to come in and seal that gap. And it seals the gap by forming a covalent bond, a phosphodiester bond, between this nucleotide and this nucleotide, and then it does the same thing here. So it's almost like it comes in and just spot welds this area so that it makes this all sealed up, a nice smooth piece of DNA, and everything's connected. So after DNA polymerase replaces the RNA primer with DNA, the enzyme ligase, I don't know if I said the name of it, it's called DNA ligase. It comes in and it welds the DNA together so that there's no gaps. Okay, so that brings us through step 6, and the only step that remains is the proofreading and repair step. Let's assume that all the pieces of primer are gone. Even this little piece of primer gets replaced by DNA. So when we're finished with step 6, what we have are 2 DNA molecules. They both are exactly identical. They have the same letters. Each one has an old strand and a new strand. An old original strand and a brand new strand. That's why it's called semi-conservative replication. Again, here's our kind of our overview. And if you look carefully, you'll see all of the enzymes that we talked about: helicase, single-stranded binding proteins. Here's primase creating the little piece of primer. Here's DNA polymerase building the new DNA strand. You can see this would be the leading strand. This is the lagging strand. So all the pieces are there. I like what they did here, though. They're showing you, if you were to zoom out, that really the way this works, it's not like we're starting at the very bottom of the DNA and moving our way all the way up to the top. DNA is way, way too long to do that. I mean, you know, your average chromosome has several thousand, sometimes tens of thousands, maybe hundreds of thousands of base pairs. It would take way too long to start at the bottom of a DNA molecule and work your way all the way up. So the reality is you actually have multiple replication bubbles. So just imagine this DNA molecule is hundreds of feet long. So we're only seeing one bubble. And we've got helicase moving in this direction, but on the other side of our bubble, if we could see like right here, helicase is also moving in this direction. So we're replicating to the right. We're also replicating to the left. And so you're just seeing kind of part of one of those replication bubbles. It's a little bit of a hard concept to explain, but just imagine, it's kind of like if you were going to repave I-40, which runs from Wilmington, North Carolina to, I think, San Francisco or LA, California. You don't start repaving in Wilmington and work your way westward to California. You're going to have about 400 repaving teams, and they're going to be working in different states repaving, a couple hundred miles of that interstate. And eventually they will meet up. Eventually all of those separate paving crews will join up and it'll be one giant repaved road. That's my metaphor. I don't know if that makes a whole lot of sense to you, but it's the best I could come up with on short notice. Okay, now this copying process works pretty well. This is how you copy your DNA every single time you go through the S phase, G1, S, G2. Every time you go through S, every time a cell prepares to divide, this is how you copy the DNA. And it works pretty darn well, but it's not perfect. On average, there's one mistake for every 10,000 base pairs. In other words, a mistake would be, if a T got put here instead of a G. If we had a C paired up with a T, that's a mistake. Whoops. Or sometimes it's not the wrong letter. Sometimes you might have the right letters in the right place, but sometimes two letters that are side by side, especially thymines like this, they will form what's called a thymine-thymine dimer. And these two thymines will try to bond with each other instead of bonding with their corresponding adenine. And it'll cause a weird little kink in the DNA. That's a mistake. It has to be fixed. So there's about one mistake or one error out of every 10,000 base pairs. That's just kind of an average. Lucky for you, you have a proofreading and repair mechanism. The DNA polymerase enzyme, there's actually several different versions of DNA polymerase, but one of them comes back in and literally scans up and down the DNA searching for mistakes. If two thymines are trying to stick to each other instead of sticking to the adenines, that will be detected. Or if we have an A where there should be a G, that will be detected. Usually the mistakes are found by DNA polymerase. And there's a process called excision repair where the bad DNA, the wrong DNA, is cut out. Excision means to cut out. And so there's another enzyme called nuclease that comes in. There's a bunch of different kinds of nucleases. They've identified 130 so far. But these are editing enzymes. They come in, they cut out the mistake. DNA polymerase replaces the faulty DNA with correct DNA. And then our welder enzyme, ligase, comes in and seals up the repair so that it's perfect. So here's a pretty cool close-up view. You see right here we've got a little kink in this DNA strand. This would be one of those thymine-thymine dimers where these two thymines are trying to bond with each other instead of hydrogen bonding with their adenine partners. And that's not good. And so here is the nuclease enzyme. It comes in and it makes a cut here, right where the arrows are. It makes another cut right here. And that little piece of faulty DNA is taken out. It just becomes scrap. And eventually it gets broken down by other enzymes. And then DNA polymerase comes back in and fills in that gap with brand new, fresh DNA that's correct. It doesn't have the mistake. And then the only thing left is for ligase to come in and seal up any gaps. So ligase has to make a bond here and a bond here. Right where that new DNA was put in, we have to seal those little micro gaps by forming phosphodiester bonds. And once ligase has finished welding, we now have a smooth, perfect, repaired piece of DNA. Now, how perfect is this editing process? Well, it's pretty good. Before the editing, before the repair, there's about one mistake for every 10,000 base pairs. After the repair, there's usually only about one mistake out of every billion base pairs. So most of the mistakes are fixed. And the ones that don't get fixed usually are not a problem because you got 3 billion letters on your DNA code. Most of those letters are not part of an actual gene. They're not part of the coding part of a gene. And so usually those mistakes don't cause a problem, but if it is a mistake, if you just happen to be unlucky and that one mistake out of a billion letters is in a bad spot that does affect a gene, then we call that a mutation. So that's DNA replication. I hope I haven't confused you or put you to sleep. And guys, I hope you're doing well. Wash your hands and stay healthy.